Hello friends, today in this video I am going to discuss about the MCQs on general microbiology, and this is the 15th part of the series of 500 MCQs on microbiology. So let's start the question answer session on general microbiology with detailed explanation. Okay, so the first question is size of Steli scalpel blade used for taking corneal scraping is number 10, number 13, number 15, or number 18, and the right answer is option C, that is number 15 is used for corneal scraping. Okay, and this is the process of corneal scraping by the by this kind of blade, and this is the scalpel blade. Okay, and this 15 number is used for corneal scrapping. Next one is in India, typhoid fever occurs as an endemic, outbreak, epidemic, or pandemic, and the right answer is option A, that is endemic. An endemic disease means constantly present in a population or region with relatively low spread. Okay, and the epidemic disease means sudden increase in cases spreading through a large population, and the pandemic disease means sudden increase in cases across several countries, continents, or the whole world. Okay, so that is known as pandemic disease. So next one is during rainy season, leptospirosis occurs as an epidemic, outbreak, endemic, or pandemic, and the right answer is option B, that is outbreak. And how this leptospirosis is spread? So the urine of rodent, especially rats, come to the water, and they contaminate the water, and from this contaminated water the bacterial leptospirosis come to the or infected the human body. Okay, and they cause the chest pain, difficulty in breathing, or swallowing body parts. Okay, so move on to the next question, that is infectious hepatitis as usually occur as an outbreak, endemic, epidemic, or pandemic, and the right answer is option C, that is epidemic. Next question is non-diffusible pigment is produced by all except Staphylococcus aureus, Enterobacter agglomerans, Pseudomonas aeruginosa, bacteroids, melanogenicus. Okay, and the right answer is option C, that is Pseudomonas aeruginosa. Next one is diffusible pigment is not produced by Staphylococcus citrus, Flavobacterium, Meningosepticum, or Chromobacterium violaceum, or all of the above, and the right answer is option D, all of the above are diffusible pigment. They cannot produce diffusible pigment. Okay, and the pigment production here, the some strain produce diffusible pigment, and those pigments are phycocyanin, fluorescein, or pyruvine. These are the diffusible pigments, and Pseudomonas aeruginosa produces characteristic grape-like odor and blue-green pus and colonies. Okay, so next one is in liquid medium, uniform turbidity is produced by Streptococcus, Staphylococcus, Corynebacterium diphtheria gravis, or Pseudomonas aeruginosa, and the right answer is option B, Staphylococcus. Next one is in liquid medium, granular deposit at the bottom is produced by Streptococcus, Staphylococcus, Corynebacterium diphtheria gravis, or Pseudomonas aeruginosa, and the right answer is option A, that is Streptococcus. Next one is in liquid medium, surface pellicle with granular deposit at the bottom is produced by Streptococcus, Staphylococcus, Corynebacterium diphtheria gravis, or Pseudomonas aeruginosa, and the right answer is option C, Corynebacterium diphtheria gravis. Okay, and here we can see this, here the pellicles are present. So pellicles are the thin layer of organic substances and dead bacterial cells which float at the top of the surface of the liquid broth culture. Okay, so here the pellicles formed due to the dead bacterial cells. Okay, so next one, that is in liquid medium, surface pellicle formation at the top is produced by Streptococcus, Staphylococcus, Corynebacterium diphtheria gravis, or Pseudomonas aeruginosa, and the right answer is option D, Pseudomonas aeruginosa. And here we can see this is the ring of pellicle, and here the pellicles are present. Okay, So next one is GSMOS staining is used to stain spirochetes, rickettsia, chlamydia, all of the above. And the right answer is option D, that is all of the above are used for used to stain by GSMOS staining. Okay, and this GSMOS stain is a differential stain that includes a combination of usine dye, methylene blue and azure in its composition, and specifically it binds to DNA region with high adenine thymine bonding levels and attached to phosphate groups. The main use of GSMOS stain is staining malarial parasite, okay, and it is used to obtain white blood cells. Platelets, RBCs and WBCs are differentiated by this method with nuclear and cytoplasmic morphology. In microbiology, GSMOS stain is used for staining Chlamydia trachomatis and Borrelia species. It is also used to identify chromosomal aberration and is cytogenetics, and also used for G banding. Okay, so next one is low temperature steam formaldehyde, that is LTSF sterilizer works at 60 degrees Celsius, 75 degrees Celsius, 100 degrees Celsius or 120 degrees Celsius, and the right answer is option B, that is 75 degrees Celsius. And this low temperature steam formaldehyde LTSF, that is a steam at low temperature, that is 73 degrees Celsius to 75 degrees Celsius and sub-atmospheric pressure 26 millimeter of Hg, is ineffective for bacterial spore, but it can effective to kill the vegetative bacterial cells. But mixed with formaldehyde, it becomes strongly sporocidal due to synergy with the steam. So that's why it allows an autoclave to perform at lower temperature for heat sensitive items. Okay, so next one is plastic and polythene tubes are sterilized by ethylene oxide, 2% glutaraldehyde, both of the above and none of the above, and the right answer is option C. It is both of the above, that is ethylene oxide and 2% glutaraldehyde is used to sterilize the plastic and polythene tubes. And this ethylene oxide sterilization, which is used almost exclusively to sterilize medical products that cannot be steam sterilized or sensitive to radiation. Mechanism of action is it destroy microorganisms by alkylation and cause denaturation of nucleic acid of microorganisms. At 30 degrees Celsius to 60 degrees Celsius with relative humidity above 30% and gas concentration between 200 to 800 milligram per liter for at least 3 hours is the effective concentration for ethylene oxide sterilization. And next, the 2% glutaraldehyde, in general the most appropriate high level chemical, it is the most appropriate high level chemical disinfectant, active against most vegetative bacteria including Mycobacterium tuberculosis and some viruses including HIV, fungi and bacterial spores, and can be safely used on metal instrument, rubber, plastic and porcelain. Okay, so next one is the for disinfecting working areas, percentage of sodium hypochlorite used is 0.5%, 1%, 2% or 5%. The right answer is option C, that is 2%. And the sodium hypochlorite, the mechanism of action is it is a reactive chlorine in aqueous solution, exists in two forms, hypochlorite and hypochlorous acid. State of available chlorine depends on pH of solution, that is above pH 7.6 it is mainly hypochlorite form and below this pH it is hypochlorous acid. So presence of 5% of free chlorine in sodium hypochlorite is responsible for breakdown of protein into amino groups, and the pH of commonly used sodium hypochlorite is 12 at which the hypochlorite form exists. Okay, and this hypochlorite dissolve neurotic tissues because of its high alkaline nature. So next, move on to the next question, that is Chitle forceps. Chitle forceps is disinfected by dipping for 10 minutes in 10% chlorhexidine, 1% cetrimide or both of the above or none of the above, and the right answer is option C, that is both of the above. Okay, and cetrimide is an antiseptic and disinfectant used topically for wound cleaning and the treatment of some skin disorders, and it is also used to treat minor burns and to prevent infection in cuts, small wounds, grazes, nappy rash and capped hands. And chlorhexidine is an antiseptic that fights bacteria. Chlorhexidine, that is a topical for the skin, is used to clean the skin to prevent infection that may be caused by surgery, injection or skin injury. Okay, so this is the forceps, Chitle forceps. Okay, so next one is the following is or are obligate aerobe: Vibrio cholerae, Bacillus anthracis, or both of the above, or none of the above. And the right answer is option C. Both of the above is obligate aerobe. Next one, the following is or are obligate anaerobe: Clostridium botulinum, Clostridium perfringens, Clostridium teteri, or all of the above. And the right answer is option D. All of the above are obligate anaerobes. So Fimbri can be stained negatively by phosphotungstic acid, nitrosin, Indian ink, or all of the above. And the right answer is option A, that is phosphotungstic acid. And this phosphotungstic acid is used in histology as a component for staining of cell specimens. It binds to fibrin, collagen and fiber of collective tissue and replaces the anions of dyes from these materials, selectively decoloring them. It is common negative stain for virus, nerve, polysaccharides and other biological tissue materials for imaging by transmission electron microscope. Next, the following Clostridium is aerotolerant, that is Clostridium tetani, Clostridium histolyticum, or Clostridium sporogens, or Clostridium novae. And the right answer is option B, Clostridium histolyticum. And the aerotolerant anaerobes means that can grow, that their growth is not affected by oxygen, so they can grow in presence or in absence of oxygen. Okay, example Enterococcus faecalis. Next one is growth factors of bacteria is or are nicotinic acid, pyridoxine, thymine, or all of the above. And the right answer is option D, that is all of the above. And this nicotinic acid is the vitamin B3, pyridoxine is vitamin B6, thymine is vitamin B1. So next one is the following is not a growth factor of bacteria: coenzyme 1, hemine, riboflavin, avidin. And the right answer is option D, avidin is not a growth factor of bacteria. Next one is oxidative phosphorylation is conservation of ADP and phosphate to ATP, ATP to ADP, or cyclic AMP to GMP, or cyclic GMP to AMP. And the right answer is option A. So oxidative phosphorylation is conversion of ADP and phosphate to ATP. So this process is in which ATP is formed as a result of the transfer of electron from NADH or FADH to 2O2 by a series of electron carriers, and take place in mitochondria, the major source of ATP in aerobic organisms. Next one is in anaerobiosis, final hydrogen acceptor is or are nitrate, sulfate, carbon dioxide, or all of the above. And the right answer is option D, all of the above. Next one is DNA contains thymine, cytosine, 2-deoxyribose, or all of the above. And the right answer is all of the above. So thymine, cytosine, 2-deoxyribose, all are present in DNA. So last question is a chemical disinfectant that should have rapid action, should be effective in presence of organic materials, should have minimum toxicity, or is all of the above. And the right answer is option D, that is all of the above. So these are the all 25 questions of this part. Okay, thank you for watching this video.